Today we're going to discuss about extrachromosomal inheritance or extranuclear inheritance or cytoplasmic inheritance in Botany Chapter 2 Classical Genetics. First, extrachromosomal inheritance or extranuclear inheritance or cytoplasmic inheritance means what? What it is? We know the DNA is the genetic material seen in the nucleus of the cell. Other than the nucleus, you have DNA in the cytoplasmic organelles also. So that in cytoplasmic organelles like chloroplast and mitochondria, they possesses their own DNA. That means they possesses their own genes. Okay, that is called extrachromosomal inheritance. So other than the nucleus, you have chromosomes in the cytoplasm also. That is called extrachromosomal nuclear, that is extrachromosomal inheritance or extranuclear inheritance or cytoplasmic inheritance or non-Mendelian inheritance. Why we saying this non-Mendelian inheritance means it won't follow the Mendelian inheritance. So because as we know, the genes are located on the chromosomes and these chromosomes are seen in the nucleus, which the genes are inherited to the next, that is transmitted to the next generation. This is the form, there is the formation, this is the normal one as the Mendel said. So that is Mendelian inheritance. But this extrachromosomal inheritance is not following the Mendelian inheritance. So we can name it as non-Mendelian inheritance. And these genes are seen in the cytoplasm. So we can name it as cytoplasmic inheritance. It is based on some of the views. It is based on some of the points. Independent. So independently it can transmit the genes. Okay, it can transmit the genes. And self-replicating extrachromosomal units called plasmogenes. They are located in the cytoplasmic organelles like chloroplast and mitochondria. So the genes are self-replicating. As I said in the first, they won't depend on nuclear gene, that is nucleus DNA for replication or for to inherit the characters, that is to transmit the characters from to the next generation. So it is able to replicate by its own. And for this they need the units called plasmogenes, and they are seen in the cytoplasmic organelles like mitochondria and chloroplast. As we study this cytoplasm, that is cytoplasmic organelles like chloroplast and mitochondria are self-autonomous, semi-autonomous. Semi-autonomous, they can replicate by its own because they contain their own DNA. And this we had studied in detail in 11th standard. In your 11th standard, you have the syllabus, cytoplasm and mitochondria as semi-autonomous. So they can able to self-perpetuate. They can live by its own. They may not be able to depend on the DNA of the nucleus. Okay. Next, we move on to the chloroplast inheritance. So where it is studied in 4 o'clock plant, Mirabilis jalapa. In Mirabilis jalapa, you have two types of leaves, variegated leaves. That is two types of variegated leaves. Dark green leaf plants and pale green leaf plants. So chloroplast inheritance is studied with the help of the crosses. You can see the picture provided, that is the cross was in a picture format. So you can see the pale green leaf plant as male. Is crossed with dark green leaf plant female. So what happened in F1 generation? You get dark green colored plant, that is dark green leaf plant is appeared. So this dark green leaf is the character of the maternal character. Maternal means mother character, female character. Why it appears means the female character, that is the ovum or the egg, contributes the nucleus as well as the cytoplasm. But this male contributes only the nucleus, not the cytoplasm. And this chloroplast is seen in where? It is not seen in the nucleus. It is seen in the chloroplast. That is, it is in the cytoplasm. In the chloroplast, the genes are present. And the ovum is dark green color. So the character is inherited to the F1 generation. Now we are, that is we are reciprocating the cross, that is dark green leaf plant male is crossed with pale green leaf plant female. So in F1 what it has appeared? Pale green leaf, that is dark green leaf plant male plant is crossed with pale green leaf plant female. In F1 you are getting pale green leaf plant. So the pale green is appeared from the maternal character, mother character only. The same point here. If it is reciprocal also, the character of the cytoplasm, that is the chloroplast, is inherited from the maternal character, female parent character only. It is transmitted. So Y means the genes, the chloroplast has the power to transmit the characters from mothers to the parent, that is offspring, not from the father side, that is male side, not from the male plant you are getting. From the female plant, that is plant, you are getting the characters because the female is contributing the nucleus as well as the cytoplasm. In the cytoplasm only chloroplast is there. So due to that, the chloroplast color, that is the pale green or dark green, it is inherited from the female plant that is parent only. Next, a cellular level of explanation of the variegated phenotype of the leaves in Mirabilis jalapa. So this is a variegated leaf with different shades, that is pale and green color. Both are here, but the pale color gives all are white chloroplast, and the green color gives green, that is green, all are green color chloroplast. So these two leaves, that is this type of leaf possesses both white chloroplast and all green chloroplast also. Okay. So next we move on to the mitochondrial inheritance. So that chloroplast inheritance is seen in Mirabilis jalapa, 4 o'clock plant, but the mitochondrial inheritance is seen in male sterility in pure maize, sorghum vulgare. So this is the best example for mitochondrial inheritance, and mitochondria is also present where in the cell? It is present in the cytoplasm. So it is called as cytoplasmic male sterility. So the male sterility is due to the mitochondria genes only. So we can name it as cytoplasmic male sterility. And this male sterility is also inherited means transmitted from the maternal parent only. Maternal means mother, that is female plant only. So the gene male sterility, that is in the cytoplasm male sterility is found in where? The gene for male sterility is found in mitochondria. And same way, here also two types of gene is there. Normal cytoplasm contains capital N. The capital N gene is responsible for normal cytoplasm. That means if capital N is there, male is fertile. And small capital S is there, aberrant cytoplasm. Aberrant means abnormal cytoplasm. That it is responsible for male sterility, or male plant is sterile. So we are going to see the cross here. So the plant with normal cytoplasm male N is crossed with plant with aberrant cytoplasm female S. In F1 generation, the male plant is sterile. So male sterile plant is appeared. Where the sterility has appeared? From the female, because the capital S is for aberrant cytoplasm plus male sterility. So the character or in the F1 is appeared from the female parent. So same way, the plant with, that is reciprocal, we are reciprocaling it. The plant with aberrant cytoplasm cannot be male. So without the cytoplasm, how we can say it is, that is the cytoplasm cannot be male. So since the pollen are sterile, so the plant with aberrant cytoplasm cannot be male. So since the pollen are sterile, so you are mentioning it as yes, okay, is crossed with plant with normal cytoplasm can be female. So it is denoted by, that is gene capital N. So the cross is there in Mirabilis jalapa, you get the reciprocal cross and you get the female parent character. Okay. But while reciprocaling it, we cannot able to, that is the cross is not possible. That is, we need two parents, male and female. But here the male is sterile, the pollen is sterile. How the cross will appear? So it is not possible. That is, the cross is not possible. No F1 generation is produced. Okay. So next, that is, they had discovered so many, that is, cytoplasmic has the genetic male sterility. It causes male sterility in, that is, it is common in the plant species, most of the plant species. So, and how it can be rectified, as we, that is, this for male sterility, the both the that is nuclear and cytoplasmic genes are responsible. And as we say in cytoplasm, capital N is normal and small s is sterile. And there are some genes can be able to restore the sterility, male sterility, with the help of nuclear genes. So that it can be referred as RF, the dominant RF. So the dominant RF is seen in the S, that is S sterile plant. It can able to restore the male sterility to male fertility. Okay, so we going to see in the cross. Okay, now the normal N, fertile male, and you have the recessive gene RF. Okay, small letters denotes the recessive gene. And capital S, that is sterile plant, with restorer gene, capital RF, it is dominant, is crossed in F1 from the female parent only will get the character of the. So it is S, that is aberrant cytoplasm with the restorer gene, dominant RF is seen. So the male is fertile. Why it is fertile? Due to the capital R, that is restorer gene. And again, capital, that is N, normal, that is fertile male with recessive gene is crossed with female, that is S, with restorer gene. In F1 generation, the S gene appears, that is aberrant cytoplasm with recessive RF. So this means the male is sterile because there is no capital R, dominant R is absent. So this shows if it can able to restore this capital R, F is nuclear gene. Along with the nuclear gene and the aberrant cytoplasm combines means it can able to restore it. So here the capital R is not here in this cross. So it is not coming in the F1 generation. So due to this only the aberrant cytoplasm is there. So it leads to the male sterility. So both the genes, nuclear as well as the cytoplasmic genes, are responsible for the male sterility. And they have been inherited to the offsprings also through the mitochondria. So that's why we named it as mitochondrial inheritance. And through the cytoplasm also plays an important role. So we are saying cytoplasmic inheritance. And atavism. So atavism, due to evolution, there are some changes, evolution and environmental condition, a particular character disappears, but the gene was present in an organism. Okay, and it is suppressed by another gene. Okay, for so many years after, so many generation after, the suppressed gene is exposed. The suppressed, because due to the suppressing gene is absent, the suppressed gene is exposed. That is called atavism. The best example I am here giving is, for example, think about that is in as we had studied, the human beings have tail. Okay, now there is no tail for us. Now we don't have any tail. Like monkeys, we also have tail. Okay, but now we don't have tail. But in future, due to evolutionary and some environmental condition, the tail can appear. So that time what we will say, this is an atavism character. That is an atavism being, human being. That is already present one character that for that character one gene is responsible and it is masked or suppressed by another dominant gene. So due to that the genus and didn't expose the character, and generation after generation of so many years or so many generation the character appears due to the that is the suppressing gene is absent. Due to the absence of suppressing gene, this gene is exposed. So due to this the character, that is the tail has exposed, that is called atavism. So a disappear character is exposed from after so many generation, that is called atavism. Okay, so we will see that is a gene is inactive for some period and it is activated after so many generation, that is called atavism. So I will read this. You can understand from this paragraph, that is these things what I have explained now. Atavism is a modification of biological structure whereby an ancestral trait reappears after having been lost through the evolutionary changes in the previous generations. So a lost character must reappear due to evolution or environmental condition. The evolutionary traits that have disappeared phenotypically do not necessarily disappear from an organism DNA. So phenotypically means physically the character is disappeared, but the gene was still there in the DNA. Okay. So what I am saying as an example, the tail will become in the future generation. It may occur also because the gene was present till now. It is in our character only. So for this I am saying one of the story, that is in Seventh Sense, Ezham Arivu, that is, in that they are provoking the character of Bodhidharma gene. So that is also in atavism. So many years back the gene was present in a particular person family and you are provoking the character, and after so many generations, 3 or 4 generations, you are provoking. That is a story, that is a cinema, but we can correlate because it is a biological fiction film, so we can correlate this with this atavism. Okay, so this gene sequence remain active or inactive for some period, and it going to be active in a particular generation. After many generations, in a particular character can be appeared. Okay, so such an unused gene may remain in the genotype for many generations. What I am saying, you understand now? As long as the gene remain intact, a fault in the genetic control suppressing the gene can lead to the reappearance of the character again. So the suppressing gene is absent there means, or any different recombinations occurred, the disappear character can appear. So the re-emergence of sexual reproduction in flowering plants. This is one of the example, that is reappearance, that is re-emergence of sexual reproduction in flowering plants seen in Hieracium pilosellus. That is the best example for atavism in plants. So that is they lost their sexual reproduction. So the sexual reproduction is absent in Hieracium plant, pilosella plant. You can see the picture. So this is the plant. That is, you can see the flower. That is Hieracium pilosella, Hawkweed. Okay, which the common name is Hawkweed. So it here they lost their sexual reproduction for so many generations. But now it has appeared due to evolutionary or weather changes. Now it has appeared, the sexual reproduction is re-emerged. That is re-emergence means again it occurred. So this is the character of atavism. So because that is, you can ask, that is how they reproduce. That as you said, the sexual reproduction is arrested in this plant. How the reproduction takes place? The reproduction takes place through apomixis. Apomixis means through asexual reproduction without the fertilization act. It reproduces through asexual reproduction. It reproduces. So now they had found out in Hieracia, pilosella, Hawkweed, there is a re-emergence, means the sexual reproduction is appeared, and these plants are now they reproduces asexually and sexually. That is through apomixis, and sexual reproduction also takes place. There are some of the questions related to the topic. So you can see it. What is mean by cytoplasmic inheritance? For this you have the answer in the topic. You can write. You can study that question. Bring out the inheritance of chloroplast genes with an example. That is, you can use that Mirabilis jalapa. Explain atavism with the help of Hieracium pilocellas. You can explain it. Explain the mitochondrial inheritance with the help of male sterility in pale maize. Thank you for watching the video. If you have any doubts, post your doubts in the comment box. I will clear it. If you like the video, give a thumbs up and share and subscribe to Science Easy Tech channel.